on this subject, kind of rounding it out of what might be useful for metabolic engineers, one concept is just having a smaller genome where you might know where everything, what everything is in your genome. The other has to do with where everything is. So the idea of refactoring. If you had the ability to rapidly change a genome or synthesize one from scratch, you could take what might be at the genomic level, sort of incoherent, disassembled parts of a pathway that appear in different locations throughout the genome, and you could consolidate them. So it is the case that in a lot of biosynthetic clusters, as we've seen with operons, genes that have similar purposes and pathways are sometimes grouped together. Maybe we can even say often grouped together, but not always. And especially long gene clusters, this is one for nitrogen fixation, shown as an example from Chris Voigt's lab, where, at least in this illustration, it seems like enzymes or genes that may encode enzymes or transcription factors are relatively clustered together. We can't really tell how much these gaps are. However, what is sort of implicit behind some of these statements is the idea that you don't actually need all of these genes. So they're a subset that are important. Some of them are transcription factors that might negatively regulate certain processes that you'd want turned on. And so you can do your own reorganization, even just within this gene cluster, which might help you, going back to our discussions of biosensors and circuits, rearrange them in a way that you can, when you're turning on a valve, you're actually turning it on for multiple steps, right? If you imagine a naturally occurring pathway or a pathway that you'd want to assemble, where you've got all these different pathway steps that are under the control of different transcription factors, or that are in several different places in the genome. If you want to knock out that pathway, maybe you can just knock out one of the genes. Or maybe what you'd like to do is really have them grouped together so that you can do more things that would influence the entire pathway. And so you can think of this also with the example that I initially introduced of the aldehyde reductases. There was one operon, and the other ones were just kind of scattered in other locations in the genome. So ideally, you might have an E. coli genome that's refactored, where you have all the enzymes that are capable of potentially reducing aldehydes, which is maybe a reaction that you want if you want to make alcohols, maybe a reaction that you don't want if you want to do something else with aldehydes. So it'd be nice if they were grouped together, because that's definitely, that kind of redundancy is a situation where you can't just knock out one and expect everything else downstream to turn off, since it's an example of many enzymes performing the same function. So I think that covers a number of different kinds of genomes, which was really the point of this genome engineering application section. We really emphasized things that were not simple gene knockouts or up or down regulation, because as we had spent a lot of time talking about metabolic flux analysis, I think it was clear that one might want to do genome engineering for those kind of interventions. But here we started with the discussion of different kinds of stress and product tolerance, really complex phenotypes, then the ability to retain or enable new chemistries, and why you might need to do genome engineering for that. And then this perspective, which is a little bit more speculative in terms of how this might relate to metabolic engineering, as that connection hasn't been directly made yet, but covering a lot of the ongoing efforts on the frontier of genome engineering to make genomes that are smaller, larger. I didn't actually talk about this much, but you can imagine that this is also on people's radars, which is this idea of if you start to create, if you can make, using DNA synthesis, really hybrid genomes. I mean, you kind of saw that, a teaser of that with the mycobacterium example from the Craig Venter Institute, where you can transplant in a genome that's different than what the organism already has, Presumably it's related, but imagine if it were from a different organism. In general, a lot of the interventions that can occur in synthetic biology, but particularly at the genome level, can really challenge how it is that we classify what different organisms are. And in any case, at least with all of the examples that related to codons, I hope you take away from this that the future of protein engineering can be really impacted by changes to the genetic code. So where we're going to go next is to talk about the design of new pathways, sometimes from scratch. And we'll cover topics like retrobiosynthesis and enzyme promiscuity.